Dear students, there's an unseen world in this earth, and this unseen world is not unknown to us. Do you know what this world is? This is the world of microbes. And today we are here to learn the products which are being produced from this world of microbes, and therefore a famous microbiologist who is pioneer in the development of antibiotics said, "Never underestimate the power of the microbe." Yes, these microbes are very much essential for the synthesis of a secondary metabolite or a product or chemically a substance known as antibiotics. So let us first learn what are these antibiotics. Why are they essential? The antibiotics, if you literally go into the meaning, means anti, antagonistic or against, and biotics, that means working against a biological system. Antibiotics can be defined as the chemical substances which are synthesized by the microorganisms, and it inhibits the growth of another set of microorganisms. Therefore, the antibiotics hold a very good place in the medical science, and they are used as therapeutic agents. The antibiotics can be produced by bacteria like bacillus. It can be produced by actinomycetes like Stretomyces, or it can be produced by molds like the Penicillium and Aspergillus. Now let us study or learn about the mode of action of the antibiotics, or when they are exactly produced in a microbial system. If we study the growth curve of a microbe, we find that there are four important stages. The first one is called the lag phase, the second one is called the exponential phase, the third is the stationary phase, and the fourth is the death phase. Antibiotic is a secondary metabolite. That means it is not required by the microbe for its growth and development. Rather, it is required by the microbes in order to inhibit the surrounding microflora that may hamper its own survivability. And therefore, it is synthesized as a secondary metabolite. So, all these secondary metabolites, which includes the antibiotics also, are synthesized during this stationary phase of growth. If we go on to study the main target of the antibiotic, we find that the antibiotic can be classified into 5 different classes. The first one is cell wall inhibitors that inhibits the manufacturing or the process of cell wall production. In this group, there is an antibiotic which is called penicillin, and the production of this antibiotic will be studied today. The second group is the cell membrane inhibitor, which inhibits the cell membrane and therefore hampers the permeability of the cell. The third category is the one which inhibits the replication and the transcription process. That means it inhibits the formation of DNA and RNA. And the fourth category is the one which are known as the protein synthesis inhibitors. Now, they may act on the 50S ribosomal subunit, or they may act on the 30S ribosomal subunit. One of the antibiotic which acts on 30S ribosomal subunit is streptomycin, and the production of this antibiotic streptomycin will also be discussed today in this lecture. And the metabolic pathway inhibitor, or the antibiotic of this category inhibits different metabolic pathway or essential metabolic pathway which is needed for its survivability. Now let us begin our study with the antibiotic called penicillin. Penicillin is a beta-lactam antibiotic. This is an antibiotic which is produced mainly by molds like Aspergillus and Penicillium. The penicillin is the first discovered antibiotic, and it is of different types. The main producers of penicillin is Penicillium and Aspergillus. Now, there are different species of Penicillium and Aspergillus who are able to produce this antibiotic. Like you can see over here, the Aspergillus, the structure of Aspergillus is like this, which is completely different from the structure of Penicillium. Both of them come into the category of mold. If we study the action of this antibiotic penicillin on the cell wall of the other microbe which is going to be inhibited, we find that penicillin inhibits the cell wall formation. Now, in the cell wall of gram-positive bacteria, there is a presence of a structure which is called peptidoglycan. Now, this peptidoglycan has got two basic structure. One is called NAG, N-acetyl muramic acid, and N-acetyl glucosamine, that is NAG and NAM. Now, NAG and NAM are joined together and they are again linked by cross linkages. Here you find the cross linkages over here and here. But when the penicillin antibiotic acts on this type of microbe, the cell wall formation is inhibited and the cross linkages over here are not formed, and therefore the cell wall structure is altered. Ultimately, the cell will burst and the microbe will die. So this is the mode of action of a beta-lactam antibiotic, which is a cell wall inhibitor, and this is nothing but penicillin. Coming on to the structure of the penicillin, why do we call it a beta-lactam antibiotic? If you study the structure of the penicillin, which is like this. Now here you find that there is a presence of an acyl side chain. In this acyl side chain, the R shows the variable or the variable side chain that may be exhibited. Also, there is a presence of a structure which is marked in blue, and this portion is called 6-APA or 6-amino penicillinic acid. Now, in this 6-amino penicillinic acid, there is a presence of thiazolidine ring, and also there is a presence of the beta-lactam ring. And because of this presence of the beta-lactam ring, we call penicillin as a beta-lactam antibiotic. Now, if I go to the structure of R further in detail, if the structure of R is like this, there is a presence of a benzene ring, then the antibiotic so produced is called the benzyl penicillin or penicillin G. And if the R group varies, then we have production of phenoxymethyl penicillin, which is nothing but penicillin V. So we find that if the acyl side chain varies or if the R group varies, we have production of different type of antibiotics or different type of penicillins. Therefore, what are the types of penicillin? Let us learn this. There are three important types of penicillin that is known to us. The first one is called the natural penicillin. The second one is called the biosynthetic penicillin, and the third one is called the semisynthetic penicillin. Now, what are the difference between the three types? If the penicillin fermentation is carried out without the addition of any side chain precursor or without the addition of any chemical compound, then the penicillin so produced in the broth is called natural penicillins. Now, from this natural mixture of penicillins, only benzyl penicillin is therapeutically useful, and other compounds must be removed so that you could purify this benzyl penicillin and therefore could be used in the humans, also in the animals. This is the structure of the benzyl penicillin. Coming on to the second type, which is called biosynthetic penicillin. Now, biosynthetic penicillin is a penicillin which is produced by controlled fermentation. That means you are controlling the fermentation in such a way that you synthesize only one type. You don't go in for the mixture of penicillins, but you synthesize only one type of penicillin of desired amount. That means the desired penicillin which is produced by the addition of some precursor or with the addition of some chemical substance is known as the biosynthetic penicillin. Here in this category, we have one type which is called penicillin G, which is produced mainly if the medium contains the corn steep liquor, and then you have another penicillin which is penicillin V, which is the first orally active penicillin used in the medical sciences. The side chain, that means the acyl side chain, varies depending on the carboxylic acid that is present in the fermentation media. That means the media which is going in for the fermentation, and therefore it can also decide that which type of penicillin will be produced. Coming on to the third type, which is called the semisynthetic penicillin. In semisynthetic penicillin, the biosynthetic penicillin is taken. That means penicillin G or penicillin V is taken. Now with the help of enzyme, it has been split off and you only have the structure which is 6-APA, having a beta-lactam ring and thiazolidine ring. Okay, so you don't have this acyl side chain or R group is not there. Now you take the 6-APA structure and then condense it or react it with some other fermentative or with some other derivative, and therefore the penicillin so formed is now called a semisynthetic penicillin. Therefore, semisynthetic penicillin is formed only after the biosynthetic penicillin has been synthesized. Now the common examples of this is amoxicillin, then pseudoxylin. Here you see the penicillin is being split down by the help of penicillin acylase. Now this penicillin acylase for the formation of the semisynthetic penicillin is very important. So from where will you get this penicillin acylase? The penicillin acylase has to be added in the fermentation media, and E. coli is a very good source of this penicillin acylase. If you compare the structure of the natural penicillin with the semisynthetic, you find that the beta-lactam nucleus is like this and you have the side chain like this. If the side chain, the pink box refers to this one and the blue colored ring or the derivative shows that you may have production of oxacillin, methicillin, ampicillin, ticagrelin, carbicillin. So all these are semisynthetic because the main nucleus has been retained while the side chain has been added or the derivative is being added, and you have production of the semisynthetic penicillin. And therefore we find that different type of penicillins can be manufactured in the industry. Now how do the industry produces this penicillin? That will be learned by us. So before that, first let us learn about the biosynthesis of penicillin. I go back to the structure again. If you study it carefully, we find that there is a blue colored box over here. This structure mimics the cysteine, that is amino acid. Another amino acid which mimics here is valine. So penicillin biosynthesis takes into account three different amino acids. One of them is valine, the second is cysteine, and the third one is amino-adipic acid. The biosynthesis therefore takes place by the condensation of all these three amino acids, and you have the formation of a tripeptide which is now called ACV or amino-adipic acid, cysteine and valine. This ACV tripeptide ultimately is converted to isopenicillin N, and from this isopenicillin N we may have formation of cephalosporin, which is another group of antibiotic which will not be dealt by us today. But the other group which is being manufactured here is penicillin, that is the most common type is penicillin G, which is found to be produced in Constip liquor. Now there is a very important factor which affects the synthesis of penicillin, and this factor is lysine. Yes, the lysine, which is an amino acid, is synthesized from a pathway that involves the L-alpha amino-adipic acid, one of the important condensation amino acid that is needed for the biosynthetic pathway. Now this lysine inhibits the penicillin synthesis because it is a feedback inhibitor of an enzyme which is called homocitrate synthase. This homocitrate synthase is an enzyme which is involved in the synthesis of amino-adipic acid. So if amino-adipic acid is not synthesized or it is not present in a proper amount, you will not have the synthesis of penicillin, and therefore lysine can inhibit the process of penicillin synthesis. Now going on to the steps of production. The foremost important thing for the production of penicillin is the presence of the strain or the pure culture. The pure culture means the culture of Penicillium or the culture of Aspergillus, from where you are going to synthesize the penicillin. Therefore, we say that preparation and the maintenance of the strain from where the antibiotic will be produced is very important for us, whether it is Penicillium notatum, Penicillium chrysogenum. So they should be either present in a plate or they should be present in a slant. The production starts with the slant or with the plate where you have the culture. They scrape out the culture and ultimately they put it in a sterile water. Here in the shake flask you have the sterile water. The culture is being taken and put into this shaker, and from this shaker flask they transfer it to a transfer vessel. They keep it for some time so that the cells acclimatize to that condition, and from the transfer vessel it is again transferred to a small tank which is called the seed tank. The seed tank refers to a tank where actually the inoculum is going to be prepared, because this inoculum is very important or crucial for the production of penicillin, which ultimately goes on to the main tank which is called the fermentation tank. If we go on to the detail of the fermentation tank, we find that inside the fermentation tank there is a presence of a culture media which is in a broth or in a liquid. So it's a submerged type of fermentation process. In this, the penicillium and the sugar is added from the top, and then in the media you have the penicillium culture, you have all the amino acids, the carbohydrates, the nitrogen source, everything is there in the media, and there here you can see the air supply is being given, or there may be presence of an agitator. Now this is because the aeration should be there, because if the aeration is not there, the microorganism or the penicillium and the aspergillus will not be able to grow properly, and therefore they will not reach their stationary phase. After this, you can see over here there is a jacket outside the fermentation tank. Now when the fermentation takes place, the temperature rises gradually. In order to maintain that temperature, we have a jacket where there may be a steam flow when the temperature cools down, or there may be a flow of cold water. So in order to maintain that temperature, you have that jacket in the fermentation tank, and ultimately the culture is removed when the fermentation is complete. Now after the fermentation is being complete, it is subjected to a rotary filter, because now it is going on for the purification extraction method. So you have the first extractor, then you have the purification column, after that followed by second extractor and third extractor. It is passed through the ceramic filter, and ultimately we have the formation of the crystals of penicillin which can be vacuum dried. The penicillin production plant, here you can see the picture, this is an industrial penicillin production plant. In the recovery process, we find that after the or we can say that 10 times, we find in 6 days there is a 10 times increase in the biomass of the penicillium. Remove 30% culture. After 6 days, 30% culture is being removed and 30% fresh culture is being given, and this is called the fed batch process. After this, the fermentation is being complete. We take it out, put it in a rotating filter, and after filtration, we remove the fungal cells or the molds, which is nothing but penicillium, and then take the filtrate. We then dissolve the filtrate in butyl acetate, and then potassium ions are added to precipitate the salt of penicillium or the salt of penicillin. Now we wash the precipitate, filter it, and then dry it. The penicillin so obtained is 99.5% pure at this stage. After this is being produced, we can enzymatically or chemically split this penicillin G molecule, and we can go in for the production of other semisynthetic penicillins. The second antibiotic which we are going to learn today is streptomycin. Streptomycin is basically an aminoglycoside having an amino group and glycosidic linkages. Here we find that we have presence of 2 sugars and a 6-carbon ring. Streptomycin is an inhibitor of protein synthesis. It basically inhibits the 30S ribosomal subunit, and as a result of this, you have the formation of the mRNA which is misread, and therefore the protein so synthesized is not correctly made and the halt to the protein synthesis takes place. The producer of this antibiotic streptomycin is basically Stretomyces, which is an actinomycetes. Now Stretomyces graeus and Stretomyces humidus are 2 main species of Stretomyces which are producers of the antibiotic called streptomycin. Here we see a single colony on a plate, and the vegetative structure are mycelial or thin thread-like structure which is seen through a microscope. If we go on to the biosynthesis of the streptomycin, we find that the precursor molecule is a D-glucose. From this D-glucose, we have formation of glucose 6-phosphate, and from this we have formation of myo-inositol, glucose phosphate and glucosamine 6-phosphate. The condensation of glucose 1-phosphate and myo-inositol through the formation of streptidine 6-phosphate and dehydrostreptose leads to the formation of streptidine 6-phosphate. However, from the other end, glucosamine 6-phosphate leads to the formation of N-methyl-glucosamine, and therefore streptidine 6-phosphate and N-methyl L-glucosamine condense together, and there is a formation of dihydrostreptomycin 6-phosphate, which is converted to streptomycin 6-phosphate, and after the removal of the phosphate group, we have the formation of streptomycin. If we go on to the production of the streptomycin, that can be divided into three different phases. The phase 1 depicts when we have the rapid growth of the mycelial biomass. Here you can see the mycelial biomass, the fungal biomass or the ball-like structures are produced. During this time, there is a little production of the antibiotic streptomycin. The proteolytic activity of the microbe releases ammonia to the media, and this is seen to cause a rise in the pH. In phase 2, we have additional production of the mycelium, and along with it, we find the streptomycin accumulates in the media. During this stage, the glucose along with the ammonia which was released in the earlier step has been utilized or consumed, and the pH remains fairly constant at 7.6 to 8. In phase 3, we say the process is being completed, and after phase 3, the mycelium of the Streptomyces is being separated out, and from the broth, the streptomycin is being extracted or purified. Now we go on to the extraction procedure by which the streptomycin will be purified from the culture filtrate. Now adsorption of the streptomycin for the culture filtrate on 1% activated charcoal at pH 6 to 8 take place. Now this 1% activated charcoal is volume or weight by volume. The elution of the streptomycin from the activated charcoal at 1.2% aqueous phosphoric acid at pH 1 to 2 takes place. However, if the absorption of the streptomycin on 3 to 4% activated charcoal takes place, then the elution takes place with the help of the phosphate buffer, which is maintained at a pH 7. The elution of streptomycin with anhydrous condition can also take place with acidified methanol. The evaporation of the neutralized methanol takes place, and there we ultimately make the volume to 1/8 of the original volume. The precipitation of the streptomycin can also take place after this process, where we add the different organic solvents like acetone or amyl acetate, which has got a higher boiling point. After these, we find the crystals of streptomycin sulfate has been formed, which can be further washed, dried, and then undergoes the process of lyophilization, when we obtain the lyophilized form of the streptomycin, which is 99% pure. Therefore, the unknown power of the microbes should not be neglected by us. The microbes, though, are not visible to the naked eye, but they are very important for the biotechnological industry. The biotechnological industry cannot survive without the aid of microorganisms, who are the producers of antibiotic. And without the help of antibiotic, the medical science would not be able to overcome the diseases that are occurring or will occur. And therefore, antibiotics are very important for the medical science, and on the other hand, the antibiotics are very important because the producers are the microorganisms. Thank you.